Hi, my name is Adam Carlson. I'm a healthcare equity research analyst here at ABG, and it's my pleasure to introduce the next speaker, Jonas Jarvious, who is CEO of linea. linea is a Swedish-listed diagnostics company focused on infectious diseases. It has just begun commercializing its lead asset, Aktier, a system for more rapid automated antimicrobial susceptibility testing, or AST, which allows for faster determination of the optimal antibiotics to use in septic patients. And it's done this together with its commercial partner, Thermo Fisher. I'll pass over the word to you, Jonas.
Thank you very much for that. I also have my colleague Anders Lundin here today, who's the CFO and IR for the company. You're exactly right. We're focusing on infectious diseases, and I'm going to guide you through our main products and what we have in development for the future to come. If we take the next slide first, this is our disclaimer slide in case me or Anders would make any forward-looking statements, but we'll move forward directly to the introduction of the company. So as you see, we have been working for many years with a clear focus on trying to improve infectious disease diagnostics. This is a huge unmet need and an escalating need in the world today. And so far, when we look back at this year, we had a super strong year. We had the CE-IVD, the clinical registration for our first lead product, Aktier, which I will focus my presentation around. We also could start the US clinical study to enter, of course, the US market. We then also had a very successful commercial evaluation, which was performed by our partner, Thermo Fisher Scientific, and of course supported by us. And we, of course, now have the European launch here in October. So very strong year, and we also really look forward to the next steps to come now, together with our partner, Thermo Fisher Scientific. But linea, as you will see in the presentation, is not just one product, one company. We could also announce, and we have produced some very strong data on our next product that we now have in development, which could really take the next big time, reducing time for these critically ill patients. So I'll come back to that at the very end of the presentation. But if we take the next slide, this is just an overview of us as a company. We are 160 employees and consultants in the company. We are located in Uppsala, Sweden. We have 3 sites. The 2 on the left is our R&D and commercial headquarters. We also, of course, have our marketing office where we can invite customers to perform demos on the instruments, but also train personnel from Thermo Fisher Scientific. On the right-hand side, you can see our production facility for consumables. As you know, this is really a consumable-driven business model, and we house all consumable production in-house. So if we then move forward in the presentation, these are the 2 topics really. Sepsis being the first application where we have launched a product towards, and sepsis is really the own immune system overreacting to an infection in the body. It's a very severe syndrome, and it's very rapidly escalating if you are not on the right treatment. And the treatment today is to fight the underlying infection, and you use antibiotics to do so. If you look at sepsis, it's the leading cause of death in our hospitals. It's the #1 cost driver in the US healthcare system. It kills around 500,000 people annually in Europe and US alone. And if we look worldwide, a person will die every 3rd second. So it's really a paramount problem we have in front of us. But where does diagnostic come in? Well, first of all, the way we treat these patients today is that we start them on empiric therapy after we place the diagnosis, and that's multiple drugs, broad-spectrum antibiotics that we initially start treatment with. That treatment is around, in 50% of the cases, actually the wrong treatment. It can be too broad or it can be too narrow. But the real problem is also that around 20% of the patients, when we start treating them with broad-spectrum antibiotics, they will still have died before you get the traditional result, the susceptibility test, because it simply takes too long. And this is what we would like to change within linea to provide that treatment recommendation much, much faster than what you can do today. If we then look at the right of the image, we can see really the next big hurdle we have. It has been posed as the biggest threat to mankind. And I'm talking about antimicrobial resistance. We have been using antibiotics for a long period of time, but of course, bacteria evolve more and more intricate ways to evade these drugs and become more and more resistant. And this is really a huge problem, and we need better diagnostics to provide the correct narrow spectrum treatment. And we see also that rapid diagnostics is needed in many, many cases, not just sepsis. If we take, for instance, respiratory tract syndromes, we can see that around 60% or even more are unnecessary use of antibiotics. You might have a viral infection, but you still use antibiotics, and that is, of course, inappropriate. So with correct diagnostics, you can also use antibiotics more appropriate to treat the patients with. And if we then look into the future, if we don't make any change on how we prescribe antibiotics, how we use it, for instance, for animal health care and so on, and if we don't have better diagnostics, it's really a very gloom-looking picture we have in front of us. It's anticipated that in only 30 years, around 10 million people will die from untreatable infections. Of course, we as a company cannot solve this entire puzzle, but we hope to be an important part, an important piece of that puzzle. So if we then move on into the presentation. So as we heard initially, we are having a worldwide partner in Thermo Fisher Scientific. It's a strong company, a very reputable company, and we have now moved to the European launch together with Thermo Fisher Scientific. There are some key aspects of the partnership that we have been able to disclose. It's worldwide exclusive for ASTar, apart from Sweden, where we also sell the product. But it's very important that we share customer data. So any customer to Thermo Fisher becomes fully open to us. We can continue to develop the product together with customers, so we don't lose that connection. A very central part of the agreement, in my opinion. It's also mutual exclusivity for Rapid AST. So Thermo Fisher Scientific have chosen us as a partner and we them. And really what's ongoing now is, of course, European launch. And we, of course, continue to train personnel, service engineer, field application specialists. So it's really an exciting time. I mean, working for so many years and now seeing the product coming out to market. If we then take the next slide. This is an image of the current diagnostic workflow for septic patients. And you can see that on the upper panel of the picture. We don't have the time to go into the detail, but as you can see, it's a multi-day process, and every circle means that you have to do something with the sample at the lab. So it's also a multi-step and quite labor-intense process. And as I mentioned, you start therapy, of course, immediately, and then you can wait 3 to 4 days before you get the traditional results. And around 20% of the patients will have died at that time point. So if we focus on the lower part of the image, that's where really Q-linea and ASTar comes into play. We can dramatically reduce time to provide the treatment recommendation, but we can also dramatically reduce the effort that needs to be done at the lab to treat the patients. So it takes only 2 minutes to start the test, and within 6 hours, you have a very broad answer on what you should use to treat the patient with. We also have a very broad panel. I will come back to that, meaning we cover a lot of different antibiotics. And that also means that you can start the AST test, the susceptibility test, at the same time as the identity test, meaning telling what bacteria is causing the infection. You need both these answers, and you can link them up in ASTar. So that, of course, also saves a lot of time to result. So if we then move on in the presentation. There are different health economic studies that have looked into the potential benefits, or actually the benefits, of having faster diagnostic for these patients. And it's quite dramatic effects. If we start from left to right, we can see a dramatic reduction in mortality if you know what you should use to treat the patient with 24 hours faster. But also, as you can see in the middle, if we can go from this broad spectrum treatment to a narrow spectrum treatment faster, we also reduce the buildup of resistance in the patient, the hospital, and in the society. Of course, very, very critically important. And if we move to the right, we can also see that the septic patients, when you progress in sepsis to septic shock, you need to be cared at the intensive care unit. They are expensive beds, and as we have seen in the pandemic, we have too few of these beds. So you could save on average 2 days in intensive care with faster diagnostics. And of course, ASTar can provide in the order of 24 to 40 hours faster. So very important benefits for the patient, the hospital, and the society with these type of tests. And of course, we're now planning to do our own health economic studies dedicated to ASTar. Take the next slide. This is just one example of the difference that rapid diagnostic provide. This is patient data from a preclinical study we did together with the University Hospital in Uppsala. It was an elderly gentleman who had neurosurgery, and then he was diagnosed with aspiration pneumonia. And in the middle part, you can see the time and all the stamps that you can see the changes or information received on that patient during these 3 days. And of course, on day 2, you got a positive blood culture, meaning that you can see that the patient had bacteria in the bloodstream. You could then start an ASTar test, and we could on the same day say that this particular bacteria was resistant towards the drug this patient was prescribed. And that was 1 day faster than the traditional care. So of course, being able to act upon this, when we know that sepsis mortality increases with close to 8% every hour when you're on the wrong treatment, that's important. We could also provide data on 10 more antibiotics compared to the standard care. So you could really make a precise pinpointed treatment for this particular patient. If we then take the next slide. So when we look at the market today, we basically look at susceptibility testing in 3 major categories. From left to right, we have the fully automated, the highest value. Then we have the semi-automated, where the lab still needs to perform some manual work, but you have a rapid readout. And then we have the traditional market. And we have built a product where we think is very competitive in all of these segments, actually. But of course, our focus is fully automated, the highest benefit to the patient and to the hospital. And if we take the next slide, we can see how we have designed ASTar. We have been working with customers really from day 1. And these are the 4 pillars that we designed the system. Should be easy to use, fast, of course, obviously. It needs to be comprehensive and accurate. And of course, when we take all this in consideration, fast is obvious, easy to use means that you can do the test by anyone at the lab at any given time point. You don't need to have higher skilled personnel to be able to perform the test. Anyone can really do it. And when we look at the comprehensive type of test, it means that you can act upon as many patient samples as you put into the system. Of course, when you invest in new technology, you would like to be able to make sure that it pays back in so to speak, that you can act upon basically any sample you put into it. But of course, you need to trust the results. It needs to be accurate. It needs to provide what we can do is the highest precision in any type of test in susceptibility, and that's the MIC value. That's the concentration that kill or inhibit bacterial growth, and we can do that with a very high reproducibility. If you take the next slide, we can really see the ease of use. This is just one user testimonial from the University Hospital in Uppsala, and they really felt that since it takes only 2 minutes to start the test, you could really free up time for the staff to do other important tasks in the lab, but still get a faster time to result for this critical patient group. So I think we have done very well in developing a super simple test for the lab to integrate in their workflow. We then take the next slide. If you look at the next part, you need to be comprehensive and accurate. This is data from our clinical study in Europe. And you can, on the left, you can see the 3 categories which you are sort of measured up against when you want to have or sell a product on the market. And it's 3 categories. Essential agreement. That means that we provide the same result, the same concentration that kill or inhibit bacteria as the reference. You need to be above 90%, and we were well over 94%, close to 95%. Then we have categorical agreement. That's a 3-graded scale, where you divide the response of the bacteria to the antibiotic in sensitive, intermediate, or resistant toward that specific drug. Again, you need to be above 90%, and we were well over 97%, close to 98%. So very, very strong data. And then lastly, you also need to be reproducible, produce the same results over and over again. Here the requirements are a bit stricter. You need to be above 95%, but we were close to 100% in this study. And if we then look at the right part, to be comprehensive, if we look at our panel as we have launched today, it actually provides the broadest combination of antibiotics and concentration ranges of any test on the market, not just fully automated rapid susceptibility test, but actually any test on the market. And as you can see on the graph, when we compare it with the other 2 companies on the market with fully automated rapid AST test, we are truly more comprehensive, providing a broader result. And of course, this means you can act upon more patients and provide more treatment guidelines. So if we then take the next slide. Another thing that we did with ASTar is we built it for the medium-sized and up hospital. These are the hospitals that perform the majority of blood culture testing in Europe and US. So we wanted to build a system with high capacity and high throughput. So you can have 12 patient samples running the system at any given time point. It's a random access-based system, so if you have free capacity, you can load the sample immediately. So a very high throughput, and of course, a goal here is to have a high pull-through in the system because the revenue is primarily driven by the consumables, of course. If we then move on in the presentation, when we look at the market for these type of tests, I mean, for blood alone, we see well over $1 billion addressable market, primarily Europe and US. But now, of course, with Thermo Fisher Scientific as a worldwide partner, we're really looking into other market segments as well. But we built ASTar not just to handle blood. We have already presented data on urine and on isolates. So when we look at the overall coverage, we see a very interesting market opportunity here. And it's really a young field when it comes to rapid testing. And I think we are really in the right timing with our product to the market where we are right now. So if we then move on, I mentioned Q-linea is not just susceptibility testing. I mean, we spent so many hours in the lab trying to understand what are the bottlenecks, what's really the driver for time to result for these patients. And what we realized is that when we now dramatically can reduce the time for AST testing, actually the next bottleneck is the transportation of the sample from the site of sampling to the central lab, where you start incubating the sample. So what if you can have a product that can use all that time for the diagnostic workflow instead of waste that time for transportation? And even furthermore, if you can do that, you can also provide equal and better care for everyone. Doesn't matter if you're sick in a small hospital or in the night or in the weekend, you can still have the same rapid time to result. And this is a product we have been developing now. And if you take the next slide, we can see why is this important. Well, this is just research articles that have looked into how long time does it actually take for the sample to arrive at the central lab. On the top part, you can see it depending on when you take the sample, either during daytime or during night or afternoon hours or weekends. And you can see that it's a dramatic time sort of added when you take the sample on a weekend. And of course, these patients are equally sick in the weekend as in the daytime. And on the lower part, you can also see the time it takes depending on how far away from the central lab the sample is being taken. And of course, we're talking about 10, 12, 15 hours. And for a critically ill septic patients, this is time you really want to use. So we take the next slide. This provides data where we are today. We are in the middle of the development of our portable culturing technology. It's called Podler, and it's basically a small thermos. So you take the sample, you put it in this container, and then it immediately starts incubate and read bacterial growth. So you can use the entire time in the transportation to the central lab. And actually, when we compare our technology with the best-in-class traditional culture cabinets, we are on par or up to an hour faster on reading or detecting the growth cycle. So apart from using transportation time, we can also actually outperform the current cabinets on just the readout cycle as well. So super interesting development. And if we take the next slide, we also think it's a super interesting market. I mean, we know in Europe and US, we have around 160 million blood cultures annually. That corresponds to a billion population, as I mentioned, 500,000 deaths. And only in that market, we see a well over $1.5 billion addressable market opportunity. But of course, this technology can be used outside Europe and US. And of course, we're also looking into other geographies when we're coming closer to launch this product. So we then take the next slide. What we have seen and talked a little bit about is ASTar, of course, and Podler, our culturing technology. But where I think Q-linea really stands out in the long term is that we actually have technology from A to Z in the diagnostic workflow for these patients. We start with blood culturing. I already talked about the Podler technology. When we look at identification, we have an absolute world-class identification system that can actually do identification direct from venous blood, I mean, without requiring a positive blood culture. We proved that technology on patient samples, but we have now focused our first product, ASTar, as you can see on the susceptibility category, to be the first product to market. But also in the susceptibility category, we have another technology called Astrea, and that's really a point of care type technology focused on urinary tract samples, so non-critical UTIs. And we've also demonstrated data to be able to categorize that within 45 minutes. So of course, our real focus now is to get a successful launch of ASTar, but we are really a company that see we have many things in the pipeline for the future. And if we look at that on the next slide, it really highlights the way we think about the future. Of course, ASTar, we want to extend the capabilities. We want to move into other type of samples, such as isolates, urine, and so on. The Podler, of course, is moving that into clinical studies and market launch. And then we have the other two platforms, Astrea and Astrid. That's more than a 3 to 5 year term, so it's longer term. But we have already proven the technology and really would like to see the market mature when we think about starting development of these products. So with that, if we take the next slide, I would just like to thank you and open up for questions. I hope you had an interesting listen in here to my presentation. I'll be happy to answer any questions. Thank you very much.
Perfect. Thank you very much, Jonas, for that presentation. A few questions. First one, if I could, on kind of the customer feedback that you've gotten so far. The system's been out in various labs now for some time. What have you heard from? I guess if you kind of divide it into kind of the lab personnel and physicians and potentially even payers, what are you hearing?
I mean, so far, I think we really are strong and positive. I mean, we designed the system to be easy to use, and we really get the response that they almost surprised how easy it is to use. I mean, it basically takes very little training. They can start to test. Anyone can do it. And of course, I mean, I would say especially now when these labs are still doing a lot of COVID testing, I mean, they're overwhelmed with job. And then you have a positive blood culture, which is the #1 priority in the microbiology lab. To be able to run that so easily is something we get a very positive feedback on. The second part we see is also the breadth of the panel. I mean, we have now already cases where it's clear that ASTar has changed treatment and potentially outcome for a number of patients because you provide a broad patient panel and very rapidly. So we also get the traction for the physicians, the one that order the test, that now realize that I can actually act upon these results much faster than I could do traditionally. So I would say so far, all the feedback we have seen is super positive in regard of use from the lab and also what type of panel and answer we provide to the clinicians.
Okay.
So far, I think it's great. We have not so much response from the economics department yet. That's more on the Thermo Fisher side, but I'm sure that will progress as well.
Okay, great. And a question on the launch trajectory. You've had a first few quarters of sales, but Thermo Fisher kind of officially launched their kind of sales campaign or so quite recently. What difference does that make in terms of the kind of the cadence of the launch? Is it still a bit too early to kind of say very much about kind of the trajectory we can expect in the near term, or what's your current thinking?
Yes, I think it's a little bit too early. I mean, one have to realize that these products are, I mean, they're still a sort of an investment to the lab. And I think we have to wait maybe 6 more months to be able to see the trajectory and plan for it to move ahead. But so far, I think we have seen very positive response. And what we could announce just basically a week after the official launch, we actually had our first evaluation system that is now currently ongoing. So I think the interest is clear there, but I think we need to wait a little bit more to see the traction build up. So really interesting time, of course, but I think we need to wait a little bit.
Okay. And a question on Accelerate. They kind of paved the way in some sense for this field, but kind of objectively haven't been able to capitalize on their first mover advantage all that well. Kind of beyond obviously designing ASTar in a different way with different features and so on. What have you kind of learned from what they've done and kind of the potential mistakes they've made, and how are you thinking about the development and commercialization going forward in terms of what to do and what not to do?
I mean, on short term, of course, we designed the product very differently. And I think, of course, based a little bit on lessons learned from their early experience, I think we have a product that's more suited for the labs and provide a broader answer. So, of course, that's maybe the obvious. I think then on the launch phase, I mean, what we have been saying also before is that this is still a young market. And I think that to be able to be long-term successful, I think you should take it in steps. You should really focus on the first stage of launch to go to the key opinion leaders, the one that will drive the field for the future. Focus on those labs, focus on perhaps the high volume labs, and then to be able to gain traction on sort of broadening the launch to smaller labs and then go extended. So I think that is a lesson learned, that this product needs to be launched carefully and with good quality. And I think that that's really the key, because we are really building our system to be transformative in this field within the 3 to 5 year scenario. So we need to make it right sort of from start. And I think that's a little bit different strategy perhaps towards Accelerate. I think also the way we work with demo instruments is also a little bit different. So there are a couple of lessons learned, maybe not all that I will share in this presentation, of course. We like to hold them to ourselves, but overall, I would say on the high point, those are the key changes. Of course, the product is very different.
Okay, good. In the interest of time, we have to move on, I'm afraid. But thank you very much, Jonas, for a very interesting presentation and Q&A.
Thank you very much.
Thank you.
Bye, everyone.